I came from Miami.
Oh, you're out of Miami. Fantastic. Well, weather's pretty nice there. I'm sure it's better here too also. So, tell us about what you're working on these days, and we're talking about lysosomes today.
Good afternoon. My name is Kong Do. I'm a biochemist by training. I spent 17 years at a consulting firm, McKinsey Company, where I helped build the healthcare practice there. Since then, I've been a serial chief strategy officer for large companies, including Merck and all of Samsung, where, among other things, I've helped start and build the biotech business for Samsung. Right? I lead a team that's affectionately known in industry as lysosome nerds. We may not know a lot of things, but we are steeped in lysosomes. My co-founder is Stuart Kornfeld, who spent more than 5 decades of his life working on the molecule that we'll be discussing today. My brother, who's truly the brilliant one in the family, he has worked on every single drug for a condition known as Pompe disease that has ever been attempted. He and I started 3 companies together. This is our 3rd company. Our first company, we discovered a drug for Pompe disease. We sold the company, had $130 million exit. We reinvested much of that into building this current company. And the rest of the team here essentially are the who's in the world of lysosome preclinical and clinical research. So first, what are lysosomes, right? Lysosomes are known as the recycling centers in the cell. So this is where stuff goes to be broken down. So just think about your own homes for a minute. What happens if you don't take the trash out on a regular basis? Things can get pretty grim pretty quickly, right? So the same thing happens in every cell in our body. As the cell grows, it creates waste, and the waste needs to go to the lysosome where they are recycled, right? And through a genetic mutation, one or more of the 50 or so lysosomal enzymes that work within the lysosomes could become inactivated. So as a result, the stuff that it's supposed to be breaking down ends up accumulating in the lysosomes. It engorges the cell, leads to organ failure, muscle failure, and kids die of excruciating pain at an early age. And there are no cures for it right now, and only about 10 of these diseases out of about 60, only about 10 of these diseases are currently treated through something called enzyme replacement therapy. In much the same way that you give insulin to a type 1 diabetic to replace what's missing in that body, you give enzyme replacement therapy to replace the missing enzyme. The key to enzyme replacement therapy is a sugar called mannose-6-phosphate. The whole field of lysosomal storage disorders all depends on sugars, particularly mannose-6-phosphate, or M6P. M6P is the natural ligand that binds to the cell surface receptor and then brings it to the lysosome where it's needed. Right? So if an enzyme does not have M6P on it, it just wanders around in blood aimlessly until it's cleared, and it does no work. So it's obvious what you want to do. You want to put as much M6P onto all of these enzymes as you possibly can, so that as much of it gets to lysosome. But unfortunately, it's much easier said than done, and no one has figured it out until Stuart a few years ago. And the reason for it all has to do with this molecule, a terribly long name, something called glucanat-1 phosphotransferase, or PTAS for short. PTAS's entire role in life is that it puts M6P, it makes M6P, it puts a phosphate onto the mannose backbone that's on the surface of the enzyme so that you make M6P, right? And this has to be done inside the Gaucher in every cell of our body. And no one has ever figured out a way to make this thing work because PTAS is a monster of a molecule. So at the very top, this is the structure of PTAS, the base pair, right? It's in the real world, it's known as an inactive zymogen that's encoded for by 3 separate genes. And it has to be cleaved at just the right spot at the 928, between the 928th and 929th position, so that it can get into its correct shape to do its work. People and companies have tried for decades to regulate this, and no one could figure it out. No one could figure out how do you cleave at the 920th position in the Gaucher of every cell in the body. So everybody gave up long ago and basically said it could never be done. But Stewart never gave up, right? And Stewart and his postdoc engineered a version of PTase that we call S1, S3. They put in some great science, brute force, and got some luck to engineer a variant of PTase that we call S1, S3 that has exactly the same shape as PTase itself. And because it's engineered not to need this cleaving, it is actually 20 times more active in making M6P than the natural PTase. So the moment I heard about this, I made a beeline out to Washington University. I convinced Stewart to join me in M6P. I hired the postdoc, licensed the patent from WashU, and essentially built the company around this core innovation. And using that core innovation, we pushed it further along. We created new technologies and so forth. And we have now created a very deep pipeline of both enzyme replacement therapies and gene therapies. Right? Note that so far we have received 6 rare pediatric drug designations. Right? The importance of this is that when we launch, let's say, this drug for Gaucher disease or this drug for Pompe, the FDA will hand us what's called an accelerated review voucher. And those vouchers can be resold on the market right now for about $200 to $300 million a piece. Right? And for any one of these diseases, it takes us less than $100 million to bring it to the market. And we will get, just from the rare pediatric drug designation alone, much more than that. Our current drug for Gaucher disease right now is ready to go into the clinic. And our drug for Pompe can be, in fact, I'm going to China in a couple of weeks and then to Australia. We're going to figure out a way to get that into the clinic in a faster way in about a year, right? For a much less cost than what we originally are planning. So we can go into any of these you want, but I like to focus first on Pompe disease. Pompe is known as a form of muscular dystrophy that is characterized by the accumulation of glycogen in muscle cells, because the body's own GAA, the enzyme GAA that's supposed to break down glycogen in these cells, are ineffective, right? So you can see the glycogen buildup in these cells compared to the normal cells. There are 3 approved drugs to treat Pompe. My brother has worked on all 3. And the standard of care remains the drug called Myozyme or Lumizyme, depending on where in the world you are. That drug is okay in keeping kids alive, right? I can look at Megan, I can look at Patrick, I can look at these individual patients who have Pompe disease, and they are alive on these drugs. But the problem is not enough of the drug gets to the muscles or around the body where they're needed. So as a result, by the time that these patients are teenagers, they're in wheelchairs and they're on ventilators because not enough drug gets to the muscle. And that's the problem that we solve. Because we have M6P, we can bring the drug to all of the muscles around the body. And what we have been able to show on the left-hand side, this looks at the level of accumulated glycogen in cells. The more accumulated glycogen you have, the more muscle, the cell damage you have. So high is bad, right? So what you really want is to get it down as low as possible. So the 3 first column here are the 3 approved drugs. And our drug candidates, M021, and you can see that we're able to get accumulated glycogen down to the level of normal mice. This has never been seen before. But more importantly, when you look at the muscle strength, that's what we're trying to do. So we're trying to help address muscle strength. This is the only drug that has ever been able to show that it can restore muscle function, right? So the black is the muscle strength of normal mice. The red is the muscle strength of diseased mice. The green are the diseased mice treated with Lumizyme. So you see that these mice do get better. but they never get to be normal. This is exactly the reason why these teenagers end up in wheelchairs and ventilators. It's they keep them alive, but not enough gets to muscles. M021 is the only drug that we have ever seen that after 2 to 3 months of treatment, we restore muscle function to that of normal mice. And then the other thing is that all of these treatments are administered through an IV infusion every other week, and it takes up to 5 hours per infusion. If you're a child in school, if you're an adult working, just imagine what that does to your routine, right? Our version here can be dosed once a month, which is a big lifesaver, life changer for these patients. And on top of it, our cost of goods is 1/10 the cost of goods of the competing drugs, right? And we have seen this pattern over and over again through the other diseases that we've seen. So we believe because of this, I can keep kids out of wheelchairs and off of ventilators. And with other data we've seen, we have a good chance of getting some of those that are in ventilators and wheelchairs off of them. Right? But we did not stop here. A few years back, my brother and I started over a dinner conversation asking ourselves a question. If we can put M6P on any enzyme to get it to lysosome, why can't we put it on any protein and get it to lysosomes as well? And that's exactly what we figured out to do, starting with antibodies, right? Antibodies generally are shaped like a Y. What we figured out a way to do is to put M6P at the bottom of that Y, right? And as a result, we can take this antibody to the lysosomes where they can be degraded. So one application in oncology, let me just give an example. When I first arrived at Merck as the chief strategy officer for Merck, Merck wanted to exit oncology. Okay. Merck wanted to exit oncology. The head of R&D wanted to get rid of this tiny little program that came in as part of an acquisition that no one understood. Right? I looked at it and I said, this will never happen under my watch. Right? Orchestrated a big fight at the executive committee about the future direction of Merck, all of that stuff. But the company changed direction, right? So that tiny program that I fought for became the drug Keytruda, right? The prior speaker spoke about. It sold nearly $30 billion last year. I'm very, very proud of that drug. Not because of the sales, my role in it, and so forth. I'm very proud of the fact that it's helping a lot of patients, keeping them alive. We can make it better. Because of what we do, we can help clear things from tumor surfaces. So on the surface of many tumor cells is expressed something called the program PD-L1, right? And so what we have done is we've created a version of an antibody that binds to PD-L1 that has M6P on it. So the antibody binds to PD-L1, M6P binds to the cation independent mannose-6-phosphate receptor that's on the surface of all these tumors. The entire antigen antibody complex then gets brought to the lysosomes for degradation, right? So we are not inhibiting the checkpoint inhibition, we're eliminating it altogether, right? So we have shown that we can remove all of the PD-L1 from the surface of tumor cells. But we then also took it one step further. Keytruda is the biggest checkpoint inhibitor out there. So we've created a version of Keytruda that essentially has the same characteristics in how it binds to tumors. We can remove all of the PD-1 from the surface of T cells, right? Again, we're eliminating the checkpoint inhibition, right? So by doing so, we are freeing up the tumor cells, the T cells, from killing the tumors. And as a result of that, we are able to show that we are dramatically better than Keytruda in inhibiting tumor growth, right? And so when I showed this data to Dean Lee, who's the head of R&D at Merck back in January, he basically said, this is like one of the most interesting things he's seen in a long time. All he was really interested in is he knows that if it works with one indication, one disease, it will work with many others. And he was just waiting for the animal data to come. So this is the first piece of animal data that we are developing. It will probably take us another couple of months to finish up the animal data, and Merck would be very interested. Same thing with J&J and Pfizer. We've had conversations with them, and they basically said, we're just interested, waiting for animal data. Okay. So our technology has created a very deep portfolio of IP. We have 9 patent families, 8 issued patents, and 20 underway. We're now sitting in Posanelli's office. Posanelli is actually one of my IP firm that's prosecuting the core original S1, S3 patents, right? And I think 4 of the 8 issued patents all has to do with stuff that Posanelli has helped me do around the world. Yeah. So our priorities are very clear. What we are trying to do is to advance the antibody platform as quickly as we can, because that's the area that's gotten a lot of interest from big pharma companies that we've spoken to, right? Pfizer, J&J, Merck, CG Oncology was mentioned earlier, and so forth. Our number one priority is to get the Pompe program to the clinic. I think we can get there within the next 12 months if we go and work in a place like Australia or China. Our journey is that whenever I start a company, I will fund everything myself until I'm convinced that there is a drug. So my brother and I and some friends, we put in the first $3 million. At that point, I was convinced we had a drug. So I went out and brought in some additional capital. So we raised $20 million in our Series A, funded and priced by Wuxi and Sixth Dimension Capital. We used that capital to essentially get the Gaucher program ready to go in the clinic, and we were in the midst of raising a B/crossover round when COVID hit, right? We were in discussions with Marshall Ways and a number of others, valuing the company between $500 million and $1 billion at that point in time. And when COVID hit, everything just ground to a halt. And so we've essentially been putting in bridge funding ever since. So we've raised a bridge round, an A3 round, back in October of 2022. And my brother and I will now continue to put in cash, about $1 million a quarter or so, to keep the company going. But that we've depleted much of our savings, so we'd like to get some external funding. So what we're trying to do now is to raise a $5 million bridge round to keep us going. And we are also in the midst of raising a $25 to $50 million B round right now. Okay. And so you may ask, how do we get to these numbers and valuations, so forth? That sounds awfully rich. Let me give you the comparables, right? When I sold Calidus to Amicus, we sold that for $130 million, right? And these were the other transactions in the lysosomal storage area that's been done over the years. Rare diseases is a very hot field right now. So these are the multiples and the valuations that are going on. And the fact that we're now into oncology, this is what J&J and Merck is really interested in. These are the deals that have been done within the last 12 months in the oncology field with assets that are in this comparable stage as we are. Okay. So what we're trying to do is raise $5 million by reopening our Series A3 round. These are these are preferred participating shares, and $5 million will essentially get roughly 4% of the company. It would not be out of the question that this would be a $1 billion acquisition in a couple of years. And I think that is very, and we're talking to some people who are very considering that right now. And this is the kind of returns that you can be expecting. $9 billion was essentially the NPV value of when we last valued the company, when we were taking the company through a crossover round. And this is how much of it will essentially go and support the trial. So what we're asking you to do today is actually invest not only to generate a return, which I believe we will generate a good return. So we have a track record of delivering. My first company, we delivered essentially 30x return for our earliest investors, but also invest to save lives, to improve lives. That's what you're really investing in. Think about those kids that are in wheelchairs that you can help prevent, get them out of wheelchairs. And one of these days, let's hope you do not need the drugs that we're creating. They're better versions of Keytruda, because we all need these better versions, right? So let me stop here to see if you have any questions. Thank you.
Hi, you said that you're putting your moiety at the the FC portion of antibodies, right?
Yes, yes.
So what happens to the whole dynamic of the FAB attaching to the sites, and now what is that tail on the
Sure.
FC doing actually?
That's a great question. Let me see if I can reshare and answer it this way. As you know very well, the FC region is responsible for helping it clear through certain immune cells in the body, right? Help clear the. Okay. So typically, what would happen is once it's bound here, the FC region could be binding to certain immune cells to help clear the antibody from the body, from circulation in the body. By putting the M6P at the tail end of that, what is really going on is that after the antibody binds itself to the antigen, the M6P binds to the cation independent mannose-6-phosphate receptor on the same cell surface as the tumor, right? Which then allows the entire antigen antibody complex then to be internalized into the cell and brought to the lysosome where they're degraded. So this is a lysosome-based protein degradation platform that uses every cell in the body instead of just certain immune cells.
So you're essentially ingesting the antibody.
We're ingesting the whole complex.
Okay, so working on that, why not adapt this system to peptide drugs and other protein drugs?
Great question again. Since I only had a short period of time, I only focused on this. But we've been able to show that we can do this for other tumor antigens. We've been able to do it to create a better version of Humira and other drugs to clear TNF alpha, interleukins, and so forth from blood circulation to help to treat IBS, Crohn's, arthritis. And we believe it could be used to treat autoimmune diseases as well. So think of hemophilia. Hemophilia is nothing more than the body making autoantibodies to factor VIII. And there, the autoantibody tends to focus on a small handful of epitopes. So imagine creating a protein that has those different epitopes, put M6P on it, and put it in as a therapeutic to soak up all the autoantibodies to treat an autoimmune disease.
Yeah, so you have an internal filtration system.
That's exactly what, yes. So it's a protein degradation platform to break things down. Thank you. Any other questions? The market for any one of these diseases is about 50,000 to 100,000 patients around the world. So this is an orphan drug. But collectively, across all of these orphan diseases, there's somewhere between 1.5 million to 2 million patients that are on this, right? And the worst of these diseases, kids die of excruciating pain before they're 3 months old or a year old. Right? And it can be treated. It absolutely can be treated with what we're doing here. That's the beauty about what we're doing is that they're like in Pompe, there are already 3 approved drugs. So the exact same codes, everything, it's already covered, right? And for creating new codes for new diseases, and the discussions we have so far is that people are waiting for it because the actual cost right now of trying to care for those patients are substantially more than the $500,000 or so per year treatment costs associated with these drugs. Yep. From a regulatory standpoint, this is actually a very simple process because it's already been used to approve probably 15 other drugs for the 10 approved conditions already. So this is a very well-known path. And because it's an orphan disease, we would probably need to conduct a phase 1/2 trial, probably 25, 40 patients maximum, and then a phase 3 trial of 50 to 100 maximum. Because these are very small. These diseases we can get results within a year. The endpoint, I believe, for our Pompe drug, we can see a difference within 3 months. All right.
Fantastic. Thanks so much, Quang.
All right. Thank you.
And Quang will be at dinner tonight. If you have any other questions, and he'll be here the rest of the day. So we're going to talk about more healthcare. We're lucky to have Lucas DeBree here from American Cancer Society. They have launched their own